infection, immunity, drugs and modern medicine. The human body is constantly exposed to microorganisms. Some of these microorganisms are harmless, but others can cause disease. These harmful microorganisms are called pathogens. They include bacteria, viruses, fungi, and protists. Once pathogens enter the body and begin to reproduce, they can damage cells and tissues and make a person ill. Bacteria can cause disease by producing toxins. These toxins damage body cells and tissues. Viruses work differently. They enter body cells and use them to reproduce. As they multiply, they damage or destroy the cells. Pathogens can spread in several ways. They can travel through the air in droplets when people cough or sneeze. They can spread through contaminated water or food. They can also spread through direct contact between people. The body has several ways to defend itself. The first line of defense includes physical barriers. The skin acts as a protective layer and prevents most pathogens from entering. The nose contains hairs and mucus which trap particles. The trachea and bronchi produce mucus and have tiny moving hairs that push the mucus towards the throat. This helps remove pathogens before they reach the lungs. The stomach produces acid which kills many pathogens that are swallowed. If pathogens get past these barriers, the immune system responds. White blood cells are the key part of the immune system. They can engulf pathogens and digest them. They can produce antitoxins to neutralize harmful toxins. They can also produce antibodies. Antibodies are specific to each pathogen and help identify and destroy it.
Once the body has produced the correct antibodies, it can respond faster the next time the same pathogen enters.
This is called immunity. Vaccination is a way to create immunity without causing disease. A vaccine contains a small amount of a dead or inactive pathogen. This stimulates the immune system to produce antibodies. If the real pathogen enters the body later, the immune system quickly destroys it before it can cause illness. Vaccination has been very successful in reducing the spread of many diseases. When a large number of people are vaccinated, the spread of the disease is reduced because there are fewer people who can pass it on. Drugs are used when disease cannot be prevented. Some drugs, such as painkillers, only relieve symptoms. They reduce pain or fever, but do not kill the pathogen. Antibiotics are different. They kill bacteria or stop them from growing. They are very effective against bacterial infections. However, they do not work against viruses. This is because viruses reproduce inside human cells, making it difficult to target them without damaging the body. One major problem with antibiotics is resistance. Bacteria can change and become resistant to a drug. When antibiotics are used, non-resistant bacteria are killed, but resistant bacteria survive. These bacteria then reproduce and spread, making the infection harder to treat. To slow down resistance, antibiotics must be used carefully. Doctors avoid prescribing them unnecessarily, and patients must complete the full course of treatment. Drugs have been developed in different ways. In the past, many drugs came from plants and microorganisms. For example, aspirin was developed from a chemical found in plants, and penicillin was discovered from mold. Today, most drugs are produced in laboratories by chemists, although some still come from natural sources. Before any drug can be used, it must be tested carefully. This is to make sure it is safe and effective. Testing begins in the laboratory using cells and tissues. Then, the drug is tested on animals to check how it affects a whole organism. After this, clinical trials are carried out on humans. Clinical trials begin with healthy volunteers. Very small doses are used at first to check for side effects. If the drug is safe, it is then tested on patients who have the disease. Scientists try to find the best dose, which is the most effective dose with the fewest side effects. In these trials, placebos are often used. A placebo is a substance that looks like the drug but has no effect. This helps scientists compare results and see whether the drug actually works. Clinical trials are often double-blind. This means that neither the patient nor the doctor knows who is receiving the drug and who is receiving the placebo. This prevents bias and makes the results more reliable. Another important development in medicine is the use of monoclonal antibodies. These are special antibodies produced in laboratories. They are made by combining a white blood cell with a tumor cell. This creates a cell that can divide rapidly and produce large amounts of identical antibodies. These antibodies can be designed to bind to a specific target. For example, they can bind to cancer cells. Drugs can be attached to these antibodies so that they deliver treatment directly to the cancer cells without damaging healthy cells.
Monoclonal antibodies are also used in diagnosis.
A common example is the pregnancy test. These tests detect a hormone in urine. If the hormone is present, the test shows a positive result. They are also used in laboratories to measure the levels of chemicals in blood and to detect pathogens. However, monoclonal antibodies are not perfect. They can cause side effects such as fever or low blood pressure. This has limited their use in some treatments. Question and answer section. Question: what is a pathogen? A pathogen is a microorganism that causes disease. Question: how do bacteria cause disease? They produce toxins that damage body cells. Question: how do viruses cause disease? They reproduce inside cells and damage them. Question: give 2 ways pathogens can spread. Through air and through contaminated food or water. Question: how does the skin protect the body? It acts as a barrier and prevents pathogens from entering. Question: what do white blood cells do? They engulf pathogens, produce antitoxins, and produce antibodies. Question: what is immunity? It is the ability of the body to respond quickly to a pathogen after first exposure. Question: how do vaccines work? They stimulate the immune system to produce antibodies without causing disease. Question:
Why do antibiotics not work on viruses?
Because viruses reproduce inside body cells and are difficult to target without harming the body. Question: what is antibiotic resistance? It is when bacteria become resistant to antibiotics and are no longer killed by them. Question: why must patients complete a course of antibiotics? To make sure all bacteria are killed and reduce resistance. Question: what is a placebo? A substance with no active effect used to compare results in clinical trials. Question: what is a double-blind trial? A trial where neither the patient nor the doctor knows who is receiving the drug. Question: what are monoclonal antibodies? They are identical antibodies produced in the laboratory that target specific molecules. Question: give one use of monoclonal antibodies. They can be used to treat cancer or detect hormones in pregnancy tests.